The key thing in biochemistry is to link the physicochemical to the biological, not to just rush to a description of the biology. The biology happens because of the chemistry, which is obviously underpinned by physical processes. Address the question behind the question and define some of the key terms and think about what a base actually is, for example, in this question. What DNA actually is, its structure.
In this video, I'm going to show you how you should answer 3 questions that could reasonably come up in your Oxford biochemistry interview. I'll be going through a detailed explanation of how to work through the question as well as giving you the answer at the end.
One of the questions we're going to go through is, why does DNA have 4 bases? One of the things that I like to do when teaching questions is to think about the question behind the question. The reason that DNA has a specific amount of bases is related to its structure. So before this, we need to think about what is the structure of DNA. And most importantly, we need to be talking about the key features of the DNA, which is double-stranded and complementary. Okay, so one strand acts as a template for the other. Okay, so because the strands are complementary to each other, you can't have a complementary system where you only have 3 bases, for example, because A will be complementary to B, but then what's C complementary to? And if we zoom in on what actually bases are, it's one of the 3 components that makes up a nucleotide. So a nucleotide is the single repeating unit of a DNA molecule. It's the monomer of the polymer that is DNA. Okay, and so your nucleotide is made up of your phosphate sugar backbone and then your base, which is variable. Okay, our bases are A, T, C, and G. Okay, so this is the necessary context behind the question. Now, why does it have 4 bases? We already established that it needs to have basically an even number. Okay, because of the fact that it's complementary. Now, why is it 4 and not 2 and not 6? Well, in terms of 2, we can say, imagine if we had a DNA sequence that only was composed of A and T. Okay, that means that at each position, you have less variability. Okay, instead of a triplet codon system, which gives you up to 64 possibilities, you would have a triplet codon system that would give you 8 possibilities. So basically, we can rule out 2 due to low genetic variation. Or it would have to be more than a triplet codon system, or could work with more than a triplet codon system. Now, on the other hand, 6 you might think is even better, because 6 to the 3 is a very large number, which I believe is, off the top of my head, 216, I think. Doesn't really matter, but it's a large number, okay? And obviously that is a huge amount of genetic diversity potentially, all these combinations. But the problem with that is there are only 20 amino acids. So you have a lot of redundancy. Even in the current system, there's a lot of redundancy. So you can actually achieve plenty of genetic diversity with just functional diversity with just 20 amino acids, because some amino acids are polar, some are non-polar, others are charged, some are small, big. Okay, so even with 20 amino acids, you can achieve optimal genetic diversity. So the kinds of things you need to be thinking about for this question is the structure of DNA, how the genetic code actually works, so the triplet codon system, and how triplet codons code for amino acids. And it's ultimately amino acids that give proteins their function. And you want to have a range of amino acid diversity in order to get the widest range of protein functions possible. But there is a practical limit, right? You could argue that, yes, 8 or 10 bases is even more diverse, but maybe from an evolutionary point of view, only 4 bases were actually stable enough, like chemically, right? So there's also other factors like evolutionary considerations that could be important. The key tip with a question like this is to address the question behind the question and define some of the key terms and think about what a base actually is, for example, in this question, what DNA actually is, its structure. Then you address why it would have to have 4 bases. Why do proteins fold? So often the common answer that students will give is proteins fold because proteins have a function, and a function needs a specific structure. The problem with this answer is that it assumes that the protein has some kind of purpose or its own awareness of what it is. It's not. It's just a molecule. That is something we need to address in our answer. This is wrong. Yes, this is true. This is not why the protein folds. Protein folding is an energy problem. What we are comparing is the unfolded protein versus the folded protein. And basically, what we're saying is on an energy diagram, your unfolded is higher energy than your folded protein. Your folded protein is more stable. Now, why might that be? There are lots of reasons for why that's the case, but some of the basic things that you can pick out is that proteins, for example, will have lots of hydrophobic R groups. Hydrophobic R groups don't like water. Where do you think they're going to go realistically when the protein folds up? They're going to go on the inside because they want to avoid interactions with water because they can't actually do the hydrogen bonding that water likes to offer, right? So the important thing to know about this question is that proteins don't fold in a vacuum. Proteins fold in the context of water. This is what you have to remember. Everything is taking place in the context of water. So a protein will fold because of it's more energetically favorable to do so for these R groups to tuck inside away from the water, and then those hydrophilic R groups to be on the surface and interact with the water molecules. Now, there are some proteins that genuinely do not fold. Their most stable state energetically is unfolded. The reason for that is because they don't really have hydrophobic R groups. All of their R groups are hydrophilic. They like interacting with water. So it's more energetically favorable for the protein to stay unfolded because all of these can interact with the water, forming hydrogen bonds. So why do proteins fold? It's a question of energy. The key thing in biochemistry is to link the physicochemical to the biological, not to just rush to a description of the biology. The biology happens because of the chemistry, which is obviously underpinned by physical processes.
Finally, we're going to take a look at the peptide bond, the actual physical chemistry of it, and whether or not it behaves like a double bond.
So if you're asked about this, the first thing you should be doing when you hear the phrase peptide bonds, you should be drawing a dipeptide. So this should be very straightforward. And, technically, if we're going to be strict about it, this group here should be an OH, should be an O- because it will have deprotonated, and this will be protonated. So this is what it would look like in water in pH 7. This, as you can see, the peptide bond specifically is this one here. Now, we can see that is clearly a single bond, but we're told that there is no free rotation. So why might that be? We should use our parallels of double bond chemistry, where in a CC double bond, there is no free rotation because you have the pi bonding system on top of the sigma bonding head-on orbitals. You have no free rotation, okay, around this axis. There's no free rotation. There's no free rotation. So if there's no free rotation here, then we have to assume that this bond also has some kind of double bond character. Why might that be? Now, again, you have to try and visualize this molecule in 3D space. What we have here is a pi bond. Remember, a pi bond is one of the two parts that makes up a double bond, and your pi orbitals exist above and below the plane of the sigma bond. You also have a lone pair on this nitrogen. So what I want you to imagine is that this whole system is almost interacting in the same plane. Remember, around this carbon here, you've got 120 trigonal planar. This is based on your A-level and IB knowledge. Around here, you've got 107. It's your trigonal pyramidal bond geometry. However, these are actual bonds, whereas this is just a lone pair. So there is some scope for geometrical flexibility. And what happens is all these three, the pi bonds, orbitals, and the lone pair, they arrange in such a way that they overlap in space. And when they overlap in space, you have delocalization. So what happens is this lone pair can delocalize in to make a double bond, and then the double bond component gets pulled onto the oxygen as a negative charge. So what the double bond actually looks like in practice is a resonance structure where you have this alternating with this, and because of this, we can say that the double bond, this peptide bond has double bond character, meaning no free rotation. Okay, so this bond is stronger than a double bond. It's something like a 1.5 bond. Okay, so again, the key principle here is to get really detailed in the actual chemistry and structure of everything. And that wraps up the 3 questions. The key principles always are: use the key terms in the question, and don't forget the chemistry and the physics that you need to explain the biological principles. And that's it.
If you found this video useful and would like further support with your Oxford or Cambridge biosciences application, you can find me at www.elvisoxfordcoach.com. Under the free guidance tab, I've got a bunch of articles about the Oxford and Cambridge interview process, how to do well in those, and also how to write a compelling personal statement. But if you'd like more further personalized support, you can start the process by booking a free consultation with me so I can assess your needs and give you specific sessions tailored to your stage of the application process.